for a long time, we have already known that bone marrow microenvironment are very kind of still remains a key player that modulate the efficacy of all the immunotherapeutic reagents. So with a disease advantage, with the studies that advancements in this field, that I think our group and others have already identified certain player or certain cellular players, such as regular T cells or maybe other immunosuppressive cells like macrophages or myeloid cells. They can not only produce cytokines or immunomodulatory or immunosuppressive markers or proteins to further in favor of myeloma cells to grow and survive. So this will be the main focus in the immediate future, that the things we know has already certain target that play a role in this very complex bidirectional interactions between tumor cells and non-tumor cells in the bone marrow microenvironment that can regulate the immunotherapy responses in patients. So, for example, one of the targets that our lab has already been studies is the CD73, and that can, in collaboration with CD39 and the CD38, they may play a very important role for the regular T cells function that can block effector T cells cytolytic activity and cytotoxicity. So we can try to targeting these molecules and try to see in combination with existing or the novel or standard of care drugs such as proteasome inhibitor and lenalidomide or pomalidomide, eventually overcome this resistance and then try to improve the overall anti-tumor immunities. And other than that, I also want to mention LAG-3, because from our studies or also others, we also find that LAG-3 play a very important role in modulating T cells and maybe other immune cells functions. And by blocking these molecules, we also recently demonstrate that it can upregulate, and because it can correlate with the disease development. So those data are also supporting the combination immunotherapies using to combine these targeting antigen immunotherapeutic drugs with the checkpoint inhibitor drugs and try to further upregulate. And the other things I'd like to mention is up and coming new target antigens that also we will be reporting in this meeting, also in the oral presentation by Dr. Yu. I think it's abstract 031. That she's going to presenting and mention about this new target antigen that have been identified in the relapsed and the very advanced tumor is PHF19, that she has a very supporting data that also identify this molecule can play a role in downregulating CD38, this target antigen, which can maybe further provide further evidence that targeting these new molecules perhaps will help to improve the current immunotherapies. And that is very exciting because these molecules actually play a role in epigenetic pathways. So by combining those target antigens that we have already know or we are still under studies that try to identify its biological function, and this provide a new combination approaches that using these molecules with different mechanism of actions and to eventually completely upregulate the overall anti-myeloma immunity and try to prolong the disease control. And eventually, hopefully, we can cure this disease.